Hello everyone, through this video we'll see the organization of the Ig genes, that is immunoglobin gene or the antibody gene. So here is a figure of basic structure of antibody, whereas the 4 polypeptide chain are present, where 2 is light and 2 is heavy chain. So Ig genes are stored as a gene segment in germline cells, and these gene segments are spliced together into a gene during differentiation of the antibody producing plasma cells. So there are two types of family of Ig light chain and the Ig heavy chain. Light chain contains two family, Kappa and Lambda, whereas the heavy chain has only one families. Kappa chain has on chromosome number 2, while the Lambda chain on chromosome 22. Heavy chains on chromosome 14. Each family consists of its own set of both variable gene and the constant gene, but in a cell expressing an antibody, each of its chain, one light and one heavy chain, is coded by a single intact gene. So the non-coding sequence or the introns are removed in the usual way by RNA splicing. The number of V genes varies with the spatial. The table describes the component of each Ig family in human and mice. The kappa and the lambda of the light chain family contains V, J and C's gene segment, which after rearrangement encode variable region of the light chain. So first one is the kappa light chain gene. In this figure, L describes the ladder sequences, and VK genes is of 300 in number, while J kappa genes have 5 in number, and C kappa genes is of 1 in number. The kappa chain gene family consists of 300 VK gene segment, each with adjacent ladder sequences in mouse. So the kappa chain family in human is similar to that of the mouse. So the figure describes the kappa chain gene family in germline cell. So here is the figure. The second one is the lambda light chain. About 40% of human antibody have lambda light chain, and the lambda light chain also undergo variation due to the genome rearrangement during the B lymphocyte development. And in this case, the L lambda gene segment also come with the V lambda and joins to the J lambda and C lambda gene segments. So here is a figure of the lambda chain gene family in the germline cell. In this, the L lambda and the V lambda are adjacent to each other, whereas the J lambda and C lambda are adjacent to each other. In germline chromosome, the DNA sequences that import lambda light chain are present in the two distinct types of the gene segment. It is L lambda, V lambda, and J lambda and C lambda in the same sequence. So third one is the heavy chain genes. So it consists of only one family gene, and it is analogous to the kappa light chain gene. In this figure, there is so many genes involved, that is alpha, beta, gamma, epsilon and the mu. So it is composed of the LH, LV, JH and CH gene segments, but there is only one additional gene segment called D, which codes for the 2 to 13 amino acids of the variable region. With one D and one J heavy gene segment deleting the two intervening sequence of the DNA to form a continuous sequence LH, DH, DJ heavy chain, which encodes the entire heavy chain variable region. So the first the heavy chain rearrange and then the light chain genes, resulting in the formation of the single function variable region DNA sequence for the heavy chain and light single functional variable region DNA sequence for the light chain. So this is whole about the organization of the Ig genes, that is immunoglobin genes or the antibody genes. Thank you and keep watching.